Hi, in this tutorial video I will briefly go over some of the stuff that you can do with MutaFrame. Now, to try an example, click on the Load Example button, and this will quickly grab one of the proteins that we have in our database, and it will load it up. It will submit an automatic mutation. In your case, you can choose from the sequence display any mutation you want. In this case, we have N to S at position 45, and the number over here is the corresponding uniprot accession for that protein. The first thing you will see is a simple comparison of the amino acids. A little bit down you will see a similarity graph that indicates how different these amino acids are. If you click over it, it will expand and it will give you a more detailed overview of what is different and what is similar. Likewise, if you go a little bit below, you will have the overall DOGAN score for this mutation. Currently, the main algorithm that we have running behind MutaFrame is a machine learning guided algorithm called DOGAN. And if you're more interested in how the algorithm actually is working and how it is designed, you can go for the small links that we have put over here for you. And in this case, you have an overall score of 0.90 about. This is which indicates that it is malicious, deleterious. And you have again a small graph indicating the score, and if you click over this graph, it will expand and it will give you a detailed breakdown of which of the features are actually contributing or not contributing to this overall score. You can always click in the links in the vicinity to have more information about what these scores mean. A little bit below, you will have the machine learning contributions. Likewise, these are the individual contributions of each sub-feature. And again, a little bit below, we will have the raw scores of input features. These are more or less useful for bioinformaticians who want to dig deeper into how these scores are generated and where they are coming from. In the case of a medical doctor or another researcher, the first number that you will be interested in will be the overall score of the DOGAN and perhaps the amino acid similarity score. If you scroll down, you will have two graphs, a distribution graph and a heat map, and these will give you the bird's eye view on your protein. That is, they display every single mutation possible that can occur in that protein of interest. Supposing, for instance, if you have a protein of 400 residues long, for each position you can have 20 different amino acids theoretically, and that would make about 8,000 data points. And this is exactly what you're looking in here. And so a few information about this graph: when you mouse over or touch any of the bins, you will get about how many values are in that interval, the absolute value of the interval, and the percentage of values compared to the total number of values. You can change the window of this graph by clicking on the bin button. So if you go left, it makes it smaller bins. If you go right, it makes larger bins. You can, instead of looking over the windows, you can click on the mode button in the top left corner, and that will decompose the whole graph into smaller parts. Now what you can do is you can use the zoom buttons in the right top corner. You can zoom into the graph, and then you can look at individual data points. If you want to zoom out to the original position, you can always bring your mouse about the top left corner and you click on the middle button, and that will reset. To see where your mutation is, you can click on the marker button, and this will give you an indication of where the mutation lies compared to this distribution graph. In our case, we have a score of 0.90. That's the 845th entry in our database, and the mutation is N45TS. One important feature is to be able to see how many mutations or deleterious or benign mutations there are in a certain part of the protein, and we thought it would be useful to make a button for that. You click on the local button and this will bring a brush tool for you, where now you can look through different parts of your protein and see where the mutation lies, where are the mutations located on this distribution graph that are coming from that part of the protein. And you can go through this brush tool and see significant parts of your protein that are enriched in either benign or deleterious mutations. For instance, this part in this protein has so many deleterious mutations compared to benign ones, so probably this part of the protein is doing something, and stuff like that. You can elaborate this further. When you are moving over the brush tool, it will give you an indication of which residues you're looking at. The numbers that you see here are the data entries. So if there is a total of 9,500 mutations in this graph, you're looking at 2,600 till 3,300. One also important thing is that you might want to see, if you locate a certain mutation, you might want to see how significant that score is compared to the rest of the mutations. And in this case, let's say you have your original mutation here. You can click on this button on the top, and then this will bring a graph where you can now mouse over parts of your graph and see the area under curve, supposedly an alias, not exact alias, but a p-value surrogate for that mutation. So for instance, for our mutation, it's quite not very significant, but it's quite significant still. We have an area under curve of 0.085. You also have information for the standard deviation of the graph and the mean. They don't change, but the area under curve will change as you move along the graph. And this can be useful for you to indicate how significant that mutation is. Other than that, you can also change the coloring function, the zooming, and you have also a small indicator of a map. When you are moving this, you can see, you can have a sense of where you are looking at the graph. Below, you have almost the same information, but this time it's a matrix of the residue count on the x-axis and the possible amino acids that can take place of a certain residue. So this is more like a heat map. Again, when you mouse over these intervals, you will have an average score for that residue for all the 20 amino acids. What you can do is again click on the mode button, and this will decompose the whole graph for you, and now you have the individual mutations. We have a color scaling that ranges from benign, starting from blue till red, which indicates deleterious, and the same buttons are there. You can turn over the local button to look through your protein. So for instance, this region is a little bit more enriched in deleterious mutations. You will realize that is the same region. Yeah, over here, if we just zoom out, it is the same region you're looking at, where there are a lot of deleterious mutations in the distribution compared to the heat map. Again, when you mouse over these individual cells, you will get information about each cell. You can zoom over them and get more information. You cannot change the bin color. These are disabled for the heat map. And at any time during your research, you can always turn on these graphs and then try to get the data, the underlying data for each of these figures, either by clicking on the JSON button or the CSV button. So the CSV is going to give you as comma separated values, and the JSON is going to give you a JSON file. You can also try to take a snapshot. So you can get the snapshot. You can click on the snapshot button, and this will bring up a new window where you can download the whole graph as a PNG file. Now, one of the most useful things we thought is to be able to integrate all of this data with other tools that are available freely on the internet. So you have resources like Exact, UniProt, Ensemble, OMIM, and it would be, we thought, quite informative to put all these things together where you can compare them side by side. So for that, you can click on this button over here. This will expand a new small window. This is one of our side projects for MutaFrame that we've been developing for the last 6 months. We have here the first things that you can try is to try entering, for instance, a mutation. So one of the questions that I've been getting quite frequently is people have been asking me, how do we re-enter another mutation on the same protein? In that case, you'd have to go scroll up, and then you would turn off this button and then re-enter the same UniProt accession and put perhaps a different protein. With this new tool, you don't have to go through that. You can just write the mutations you want to look through, because you have the whole data here anyway. For instance, if you're looking for methionine 1,2-valine, and then you click on the label mutation button, and that will find it for you. If you zoom in this graph briefly, the after a few seconds, the label is gonna find its original point and relocate there. So you can add additional mutations, let's say S300A, I'm not sure if it's there. Okay, it's not there. It will give you a small indication of a warning. We can try, let's see another mutation. What is it? F200, F262, let's say, to arginine. In that case, it will find the mutation that you entered. Now we can freely move this graph up and down, and these will relocate. You can add as many mutations as you want. You can, if you want to clear these mutations, then you click on the clear button. If you're not looking at any of these graphs, like either the distribution or the heat map, if you're just somewhere else and then you try to label a mutation, then you will have a warning that you need to scroll down to either the distribution or the heat map. The same things I've shown you for the distribution, you can do it over here as well. So if you want to label a mutation, there you go. So you can label as many mutations as you want. One other small addition that we have this year is to start a new add-on that we call it Warcraft, which stands for Variant Craft. You click on this and it will start fetching data from different sources, including Ensemble, OMIM, Exact, etc. It will store the data in your computer. If there's not enough space, then it will make a fresh call. There are a few buttons over here. First of all, you can drag this freely around. You can resize it to your liking. If you click this button over here, it will clear it from your browser cache, so you can refetch the fresh data if any of these databases are updated. For now, I will keep it default. You have the genome build, you have the gene symbol for that corresponding UniProt accession that we had in the beginning. You have the MIM, the ESNG, the transcript, the golden transcripts accession. You have the chromosome start and values as well. And over here you will have the chromosome position, the reference allele, the alternative allele, the minus log allele frequency, the cDNA encoding, the protein annotation with HGVS, whether if it exists in the OMIM, and the Diogen score. So all these mutations that you see over here are actually taken from Exact, and they are cross-referenced with what we have in our database, OMIM, and some other sources. So how you can use this is to mouse over any area under the table, and there will be a small brief animation. So if you give it a go, about a second till the animation is complete, now you can mouse over or move your touch point. If you're trying this on a mobile device, then it will expand the part that you're looking at. And in this case, for example, I'm looking at a mutation from G to C that results in arginine to proline at residue 157. And that will also cross-locate that mutation on the distribution graph. It does not exist in OMIM here. But it does exist in our database and also does exist in the Exact. And for this mutation, we have a Diogen score of 0.48. So you can look through every mutation like this. So this mutation, for instance, has a relatively higher score of 0.86, and it does also exist in OMIM. We have a pagination system. For each page, we have about 75 mutations. So there's the second page. There is the third page, and you can also click on the all button to reveal all the mutations. For some proteins, we have a lot of mutations in Exact, and in that case, it might reduce the frame rate in your browser. So for your performance, I recommend you trying out the paginated view first. Over here, under the position part, you will have small markers that indicate where does this mutation exist with respect to the whole genome location. So you have these indicators. You can actually see the surrogate markers for the exons. So you can use this to cross-reference from different databases and see where each mutation is located. So if you're not looking at any of the distribution or the heat map, you can just expand this and then look through what mutations are there. If you go through the distribution, or if you're looking into the heat map, if you're in the vicinity of these, then when you're on this mouse over the table, when you're looking through this, it will also highlight for you where the mutation is. And this is very useful to see at the same time what is the DOGAN score, what is if it exists an OMIM, and stuff like that. So the most important thing here is that the color coding is the same for the DOGAN. The red is deleterious, blue is benign, and this color code that you see on the HGVS annotation is also the same color scheme, and these scores are coming from the first section that I've already shown you. It's the amino acid similarity overview, so you will have the same color coding there as well. So when you are, for instance, looking for a certain mutation, you have on the rightmost side the DOGAN score, and then you have also the amino acid similarity score colored from blue to benign, or blue to similar and red to different, and that is also indicated underneath here as heuristic. So the DOGAN score is 0.35 as indicated here, and then we have the heuristic score. The same thing goes for the heat map as well. So these are some of the overall new things that we have implemented, and I just want to give you an idea of what you can do with our tool. I'd be delighted to have your comments and your suggestions. You can always let us know about some features that you want us to implement via using the Discuss tool over here, or sending us an email, the Google groups, or from the YouTube channel over here. So thank you very much. Have a good day.